He kōna e purangi tēnei nā te reo irirangi o Aotearoa.
Kia ora, nau mai haramai ki te au hurihanga. Hello and welcome to Our Changing Worlds. Ko Clegg and Cannon tēnei. Earlier this year, a team of New Zealand scientists spent 3 weeks at sea exploring the Bounty Trough. That's a deep-sea canyon off the Otago Coast. And they returned with bucketfuls of specimens that are brand new to science. But that was just the beginning. These discoveries are still keeping them busy months later, and science journalist Veronika Meduna went along to their taxonomic workshops to see what lives in New Zealand's deep oceans.
There's an estimated 2 million species living in the ocean, but so far we've only identified about 10% of them. The Ocean Census, a project under the umbrella of the United Nations Ocean Decade, aims to address this. The deep underwater rift valley of the Bounty Trough was chosen because we know a little bit about its geology, but very little about the animals that live on the seafloor.
It's basically like a really big, massive valley.
Sadie Mills is a marine biologist and invertebrate collection manager at NIWA, and she was the voyage leader.
It has these slopes that slope down into a channel, something like a river channel. It's actually never been a river on land ever. It's always been a marine, oceanic kind of river. And it's like a canyon, big deep canyon walls with rocks and boulders on them and animals kind of clinging onto that, and then down into this river-like basin that then goes from about 1,500 metres at the shallow end down to 5,000 metres at the deep end.
Imagine dropping a camera down to 5,000 metres to the seafloor to see it for the first time.
We can hover the camera just about 1 to 2 metres off the seafloor and get really impressive images coming back. And we can watch that live whilst we're sitting at the top in the lab on the ship. And it's very exciting when we get that first glimpse of the seafloor. It was very dark, and then the lights turn on and we start to see creatures swimming around, the fish swimming around, and the amazing creatures sitting on the seafloor like sea stars and worms and all sorts. So yeah, it's a very special opportunity to see that.
Think ocean and you probably think fish. And indeed, the expeditioners did return with new fish species and sightings. For Te Papa's fish collection curators Andrew Stewart and Tom Linley, the deep ocean was a treasure trove.
Well, when you look at our holdings, you find that just about all of it is taken as a result of either directly the seafood industry or NIWA doing research on commercial species. So there's a drop-off of numbers of specimens and species in the collection from 1,200, 1,500 metres.
And it's not gradual. It just stops.
Just bump.
There's a shelf there. And our EEZ goes all the way to over 10,000 metres, but we've got a handful of samples from beyond those depths.
And over half our EEZ is deeper than 2,000 metres. So over half our exclusive economic zone is completely unexplored.
Andrew and Tom had joined the voyage with different types of fish traps and a hit list of what they hoped to see or catch. They shared a cabin and worked opposite shifts until one moment when Tom came banging on the cabin door.
You'd only just gone to bed. You would have been right in REM sleep.
I was in REM sleep. I'd been asleep for a couple of hours, I think, and then REM sleep, and there's this weird dream, and there's the phone is going in it. And I wake up, and the phone's still going. So I'm thinking, the trap's up. This must be good. Tom's waking me up.
He would have never forgiven me if I hadn't woken him up.
No.
That particular baited fish trap came back from 2,700 metres, and in it were long, slippery fish. We're chatting weeks later at Te Papa, but on the bench next to us is a jar filled with what prompted that cabin door banging, a specimen of eelpout.
It's watching you.
It's watching us, yes. Move across here. So this is the smallest specimen, and I quite deliberately pickled this one on the ship.
This is what we picked as our poster child. This is the one ready for media.
The eelpouts are generally not big as a group anyway. Slender and rounded bodies, as you can see. Also extremely slimy. So if you get them in a net, commercial net, there's a good chance they could just wriggle their way through the mesh and be gone. The other problem you've got with these for catching them is that submersibles and ROVs have captured pictures of them around the world, and they like associating with really rough bottoms and foul ground in amongst rocks and crevices and holes and rock jumbles. Somewhere you really don't want to be putting a net down because there's a good chance you won't be bringing it back.
They tend to be campers, don't they? They set up shop in certain places. So these are the ones you'll often see around whale falls, around the skeletons. You'll see them around hydrothermal vents. So anywhere there's a food source, they sort of set up camp, but they're benthic fish. They sit on the bottom. They don't really swim up, other than a few weird pelagic species. But they don't have a huge range, which is why.
And they're not strong swimmers.
Yeah.
There was one brief bit of film footage on the deep water camera, and there was one swimming along the seabed, and it was the clumsiest thing. So they're obviously much more at home if they can just sort of squeeze in and around and through cracks and holes and crevices and rock formations.
They seem to be secondary predators. When we did lots of baited camera work, they wouldn't really eat the bait. They would eat the things attracted to the bait. So they set up shop, they realize that this is going to be a good hunting ground, and then they snuffle up the sort of amphipods and invertebrates that come to eat the carcass. And we've even seen them squabbling with each other over that food source. So we've seen them defending it. So they obviously see it as a valuable hunting ground, and they'll defend it from other eel pouts.
As the name suggests, they do look a bit like an eel, and this helps them move around undetected.
It's a very efficient way of getting around without actually advertising your presence. If you've got an eel-like body with a long fin that you just ripple, and it's a low energy sort of budget to move, and you're not advertising your presence, you can sneak up on prey, and you're not sort of, as it were, shouting out on the savannah and raising the attraction of bigger predators.
Something like a tuna is a rocket ship. It's pushing water behind itself, whereas these, to save energy and again to be subtle, they're rolling water down their bodies, so they're only touching the same piece of water as they move along. And if that water is full of bioluminescent things that flash when you brush up against them, it's another way of hiding your presence because you're not setting off these burglar alarms. So it's more Mission Impossible laser gauntlet down there.
Yeah.
You want to sneak through without setting off the burglar alarms.
When these three specimens came up in the trap, it looked like two different species. But first they needed to figure out exactly which ones.
The humorous side of this is the world authority on this group, who's currently still active, is a guy in Copenhagen, Peter Muller. And Peter's a good friend, but I sent him an email, and then about a month later I've got to email his technician going, can you get Peter to have a look and answer that email? Well, he got back to me within 12 hours and went, I got to see it. And so I sent
X-rays, more pictures.
Yeah, more pictures. Send me the pictures. So I sent him the pictures. X-rays. And rather excitingly, he said, I think the species you're calling Pachycara might be this Antarctic genus called Sant Elmoa, which would be a major departure from the Southern Ocean. So armed with that information, I dived into the known species, and, well, if it's that genus, it's new. It doesn't fit any of the Pachycara species that we know of in this region. So whichever way you want to cut it, there are two new species there.
But they couldn't do this kind of species identification for everything, because some things remain tantalizingly out of reach.
And there was the strange bibby-esque fish that we saw twice on the camera system.
The one mocking you on the cameras.
Yes.
So that was an element of it as well. We'd send down a towed camera system before we deployed anything because we'd need to know what the bottom was like. This was unmapped area. We could go down there and it's full of gravel. We could go down there and it's cliffs. So we'd always do this recce, and the nets are probably touching the bottom for maybe 15 minutes, but these tows were maybe, with the camera system, were maybe half an hour, sometimes an hour. I think there was one really long one as well.
Yeah, hours.
So you get to see a lot more stuff, and you get tempted by what you want to come up when you put the gear down. So we saw a lot of stuff that we didn't get, and we know it's new. We know it's something weird.
Yes,
so it's a bit Christmas morning, really.
We're referring to bibbies.
Now, if you, like me, are thinking, what even is a bibby?
That's a nickname we've given a group of fishes called snailfish. So you go down through the water column and things become more and more, I'm loath to say grotesque, but more extreme.
Toothy and pointy.
Massive fangs and light organs. You're sort of thinking, as you go deeper, that just stuff gets weirder and creepier, and you get down to the bottom, and there's this thing that looks like a teddy bear. They sort of go, ooh. ooh, it's just this pink and squidgy, and
you go far enough, they go cute again.
Yeah.
You just got to go through the scary bit.
And somebody called them bibbies because they just look so ridiculous.
There's a great little cartoon that we became obsessed with of sort of saying that normal fish, nightmare zone, and then just bibby at the bottom, and yeah.
But there was one brief film clip, and I could see this little fish sitting on the bottom. It was eel-shaped. It wasn't particularly dark. It had the kind of right colour. It had the right activity. And then suddenly it becomes aware of the camera system, probably more the sort of vibrations and the water pressure coming ahead of it. And it just freaks. And it starts bouncing around and then tears off right in front of the camera. And I'm able to freeze frame it at 1 second in, just before it zips off. And Tom said, yeah, that's it. And I said, behaviour's all wrong. And then I said, there's the other thing, this thing called the tadpole slickhead. Great name. Name says it all. And I showed Tom, and he goes, it's an imposter.
Yeah, the ones that converge.
Yeah, but the problem with the bibby ID is that snailfish don't behave like that. The trouble with the tadpole slickhead ID is we're 1,000 kilometres too far south and 1,000 metres too deep. So what is it? We don't know. And we saw it twice on 2 separate occasions, reasonably well separated. So it's not just a straggler that wound up there going, how did I wind up here?
Now think ocean again, but this time think of everything else alive down there that's not a fish. Because while a number of new fish species may be countable on one hand, there is a group of deep-sea animals that will likely deliver tens of newly described species. Dr. Rachel Purd is an expert on marine invertebrates, especially small crustaceans known as amphipods and isopods. I meet her at her office at Niwa, which really looks more like a natural history museum collection. There are stacks of boxes full of jars everywhere, various microscopes on her desk, and whatever desk space remains is covered in small glass vials full of centimetre-sized amphipods.
In our group, which is called the pericarid crustaceans, and they're generally smaller crustaceans, but what holds them together is they're all brooders. So they don't have a larval stage, they just have eggs, and they hold the eggs with these brood plates under their bodies until they hatch, and then they hatch out as mini-me's of the little amphipods or isopods. And they have about 10,000 species each across the globe.
And they're all marine?
No. So the isopods you would know as like slaters and pillbugs that you have in your garden. They're crustaceans.
Or in my house.
Or in your house, under the pot plants. So that's the very typical isopod shape, that flattened slater look.
But they're also common in the marine environment.
Oh, yes. They're much more dominant in the marine environment. Pretty much once you get past 10,000 metres, there's very few fish. There's very, very rare decapods. There's the occasional prawn that gets down there, but it's dominated by amphipods.
One other thing that binds them together as a group is that they're all quite leggy.
Yes, it's in the name. So isopods and amphipods, they end in a pod, and pod is Latin for leg. And so isopod means all their legs are the same, whereas amphipods, they have legs that face forward and legs that face back.
If they're that common in the marine environment, you would have expected to find quite a lot.
Yes.
But did you see any surprises?
New Zealand's fauna for amphipods is kind of well known, but it's well known in patches. And the Bounty Trough was one of those areas that wasn't well known. We've got some material from closer to the shoreline, and when we looked at it, 80 to 90% of the amphipods were new to science. We almost expected to see that similar numbers. So that was just off Otago, so very similar area but a lot shallower.
And did you see that with the Bounty Trough?
Yes.
At the time of my visit, Rachel was already certain she had several new species, but it could be months before she's worked through all the samples from the Bounty Trough voyage.
I've divided them into families, and then within a family you have a variety of species. So I've just started looking at one family, one genus, and I found 3 new species.
How do you know? So you're looking at them under the microscope.
Yes,
So there's
and you look at all the features.
So there's no genetics, nothing
Not
internal.
yet.
It's really just
looking. Actually, taking the genetic samples so that we can verify. So I like a really integrated approach to describing a new species. There are people who just go genetics. That's it, full stop. But for amphipods, they have such a wealth of features on the outside and on their mouth parts that you can actually tell morphologically as well as molecularly.
So yes, high-tech genome sequencing is an important method for taxonomy.
I draw mine.
Rachel also still relies on some very traditional tools.
So these three are all three new species from the Bounty Trough of the same genus. So they're all really closely related. So this one had really ridged, the first few segments of its body were really bulbous and ridgy, and then the last segments were really spiky. And it still had the same long legs. This one was really smooth in its
in the front part.
Part.
Yeah.
So it wasn't this rounded blobs, and it still had some spikes, but the spikes were different. And then this one that I'm drawing now also has the smooth front part of the body, but its spikes are different.
In their jars, it's hard to tell the difference between the amphipods other than by size. But it's a very different story under the microscope. Oh, it's got some long... It's
got really long, delicate legs and some spines on its back.
And hairy legs too.
Yeah.
Could you have a look and describe it for me a bit more than just long-legged? That's what I can tell.
So, this animal has obviously lots of legs, and they're the obvious bit. And it has, each leg is divided into segments. So they're segmented leg organisms, which make them arthropods related to insects, which just means jointed leg. The head has two sets of antennae. This one doesn't have an eye, so it's a blind one, which you quite often find with deeper sea.
How deep?
This one's from about 1,400 metres.
Oh, so it's not super deep.
It's not super deep. For the Bounty Trough, they went as deep as 4,500. So this one has lots of segments. We can see that the body is segmented, but each leg is segmented. At the front end, you have the head, and then they have this bundle of mouth parts that are there to capture food any which way. So these ones will probably be opportunistic feeders, but their mouth parts often vary. So if they're herbivore, they've got big molars and big teeth for grinding away. If they're filter feeders, they'll have really delicate, lots of setae. Setae is another way of saying hair, to catch all the particles in the water. So if they're scavengers, you quite often big strong teeth and big strong claws to try and cut through bits of flesh.
Amphipods are versatile, ubiquitous and dominant in the deep ocean. And Rachel says that's why it's important to know more about them.
The Bounty Trough material has been such a great opportunity to focus on a certain area where we can actually go, these animals are there, they're transporting the carbon, they're part of the food chain, they're actually doing these really interesting things. And in a world where the climate is changing so dramatically, we need to know how the deep is related to the surface, how every organism is vital in that contribution to how healthy the ocean is.
At Te Papa, mollusk curator Kerry Walton has managed to sort through almost all the samples he brought back from the voyage. He's already identified 78 new species of snails and mussels. But that may not be the end of it. About 80% of New Zealand's marine mollusks are found nowhere else in the world. Every new discovery is precious because each mollusk shell tells a story about its own evolution and its ecosystem. Kerry shows me Te Papa's mollusk collection, a long row of tall cabinets filled with hundreds of drawers. It holds as many as 40 million mollusk specimens. Some lots represent the same species but have been collected over several decades or from different geographic regions.
When you think of mollusk, you might imagine something like a large scallop fitting in your hand, or an oyster or a paua, something that you can eat, something that you can conveniently see and something that you can find either eating the lettuce in your garden or by wandering along a beach. But the reality is the vast majority of mollusks are LBJs, little brown jobbies. A large mollusk is over 6 millimetres, and quite a few species never exceed 1 millimetre even at maturity.
New Zealand has about 6,000 mollusk species described as living animals, and a further 8 to 10,000 are present only as shells in the fossil record.
A couple of the advantages of working with mollusks is that the vast majority, perhaps around 90%, produce a shell, which has high preservation potential. And what that means is, even though we haven't found a live animal, the right habitat, we can find evidence of its presence. And the majority of mollusk shells are detailed enough. They have little structures and sculpture that we can identify them to species without needing DNA, without needing the soft tissues for anatomical work.
Te Papa already has many mollusks from the Bounty Trough, but Kerry says the voyage added invaluable new insights about the deep ocean.
Within the Bounty Trough region, we had 27,000 mollusk lots already here at Te Papa. But of those, only 1,000 were from deeper than 1,000 metres, 16 were from deeper than 2,000 metres, and we didn't have anything from deeper than 2,500 metres. So for us to have travelled the length of the Bounty Trough, working down to almost 5,000 metres, was wholly new ground for us. Despite a well-known fauna, this is somewhere we've never been. So the 78 species that we've confirmed so far as being new to science, not just new to science in the sense of being unnamed, which is actually not that remarkable for mollusks, but material that we'd never seen before. That was not an unexpected amount, but it was an amount we were quite pleased with.
As much as a third of marine mollusk species have a strong relationship with other habitat-forming organisms, such as sponges and corals. So finding a mollusk shell also reveals clues about the broader ecosystem, even if that's no longer present.
So this group we're looking at now, one of the subfamilies is strongly associated with stylasteran corals, like your red coral. And by collecting the shells, we can infer what coral species or families are present in a region. One of the seamount sites, it was mostly rubble, there were very few live corals on it, but we collected about 6 species in this group, the callistomatid snails, which can tell us that this formerly had quite a diverse coral community, and the fact that we got a large number of these would probably be evidence that this was a dense and healthy area as well.
But mollusks don't just live on corals. Some are parasitic. They live their lives feeding off other animals. For example, echinoderms like sea stars and sea cucumbers. And they can be very picky. Kerry tells me about a family of small sea snails, known as eulemids, that he finds tricky to work on because they have transparent, featureless, pale shells and tend to look very similar.
What we can tell from the ones that we can identify is what echinoderm lineages, at least, are present. Many of these parasites are only found in one species or only found in one genus, so these are one of the informative groups that can tell us a lot about the non-mollusks that are present in a region.
So the relationship can be as tight as species of mollusk with species of, say, sea star, brittle star, sea cucumber?
Absolutely. And one of the eureka moment new species was on a gummy squirrel, a purple deep sea cucumber, on which there was a rather large, by eulemid standards, eulemid parasitic snail, which is about 1 centimetre across. And this was definitely something we hadn't encountered previously in New Zealand. The most similar species we can find to it is from the Northern Hemisphere, so unlikely to be closely related. Whether or not that if we find this shell in the future, that tells us that this type of gummy squirrel is present, or gummy squirrels more generally, we don't know. We've got a sample size of 1. But that's the sort of extra information that we can gain from amassing natural history collections like these.
Mussels are perhaps less ornate and beautiful compared with snail shells, but they too tell a story about life in the deep ocean.
The mussels are an interesting group. These aren't the green shell mussels, green lip mussels you might be used to. This group here, the Bathymodioline mussels, these are from whale skulls from the deep sea. 900 metres, 700 metres, 1,200 metres.
Wow.
When we encounter whale bones in the deep sea, whale bones are exceptionally full of fats and oils. We've got specimens over in the whale collection where they've been cleaned extensively and here for over 100 years, and yet they're still oozing oil. And what that means is they release a lot of hydrogen sulfide as they break down. These mussels are quite special in that they don't have a digestive system, or they've got a greatly reduced digestive system. Instead, they've got bacteria that they farm in their gills that metabolize sugars from the hydrogen sulfide, the smelly gas that gives farts their unpleasantries. And bacteria aren't very effective or efficient feeders. For every 3 sugars they produce, they sort of let a couple go by accident. And the mussels absorb that directly. That's the tax for having a nice modified gill in which to live. They're relatively featureless or relatively character poor. They look like a brown bean, if you like. And yet we've got at least 4 or 5 genera, different major lineages within these mussels in New Zealand, and they're associated with whalebone and with decaying wood. One sample we got at over 3,000 metres on the Bounty Trough expedition on a tiny piece of bark, smaller than the size of your fist. I got a single mussel, and I'm excited to see what the DNA sequencing results for that will be, because that's from substantially deeper than any of the hundreds of lots of mussels that we have here.
All kinds of mollusks can live even deeper than the 5,000 metres this expedition sampled. But there is a limit, and it's to do with the ocean's changing chemistry.
There is a certain depth at which you can no longer effectively create carbonate of a shell, and that limits the distribution or the depth range for many mollusk species. One of the big worries with ocean acidification is that the layer at which it becomes ineffective to create carbonate, which is what many mollusks and other invertebrates rely on for their skeletons, that layer might become shallower as we change ocean temperature and ocean pH. Those are 2 of the key factors in determining at what point, at what depth, it becomes impractical to make shell. With deeper water samples, it's not unusual to have these highly eroded shells that it might have been a live-taken individual, but the shell is so eroded that it is really difficult to see any of the diagnostic characters.
There will be a process of where the mollusk still keeps producing the shell from the inside, but on the outside it keeps getting eroded.
Exactly. And we encountered a couple of false starts, if you like, on this voyage. We were towing the camera at 3,500 metres and encountered a fairly large white whelk. And I was excited because we have no whelks of that type from that depth, so this was almost certain to be something new, either a new record or a new species. And unfortunately, on our first deployment of towed gear, we didn't get any samples. So all the information I had was, well, we know there's something there that we don't know anything about. We were lucky to be able to redeploy the gear on the way back, and we managed to get a sample. And the specimen was so eroded, it was a live-taken animal, but the shell was so eroded that I couldn't see any features of it. I could conclude only still that it was a white whelk. I could guess what family it belonged in, but until we get sequence data or anatomy data, we can't be sure what it is exactly.
Discovering a new species is exciting for any biologist, but the expedition's bounty of more common species has its own value. Without knowing who's who in the deep ocean, we can't even begin to appreciate it, let alone protect it. Here's Tom Lindley and Andrew Stewart again.
We have a duty to understand the fauna of New Zealand and to have a record of the fauna of New Zealand. And so a big drive for this, as happy as it makes us and as animated as you can hear we are, it is our job to have a record of what's there. And it's difficult that because so much of our waters are deep, that we are actually locked out from exploring a lot of it. And so that's why it's so exciting to have records of these animals. So future change, future shifts, until we have a baseline, we don't have anything to measure that against. If this new species here disappeared tomorrow, we wouldn't have had a record of it. We would have never known it was there. So how can we manage that? How can we measure any changes that are taking place if we've got no baseline?
And this is the world's largest habitat by a vast margin. I mean, we arrogantly call the planet Earth. It shouldn't be. It should be called water. And more specifically, it should be called really, really deep water. This plays a massively important role in things like temperature, climate, productivity, oxygen production or not. We really have to know what's down there, because shifts down there are the canary in the coal mine. And while some people will say, well, what's its value?
Yeah, it's so far away from me, how can it impact me?
Why should I care? Life is more than just what I can eat and what can earn me $1. And the image I like to use is the Bayeux Tapestry. Nearly 1,000-year-old, 70-metre long needlework depicting the events leading up to and covering the Battle of Hastings in 1066, when William the Conqueror came and defeated Harold. Now, if you actually look at pictures of the tapestry, you've got all the sort of action happening in the words and the comets and all this sort of stuff, and all coloured threads, beautiful, but vast majority of that tapestry is white linen thread, and it's holding the interesting stuff together. No one in their right mind would think, well, let's cut out some of these white bits. They're boring. They all look the same.
They don't tell a story.
They don't tell a story. They're just taking up space. Who cares? You would very quickly become acquainted with the French legal system if you tried that stunt. And all these things that we're finding, while they're remote and they're weird and they're small, they're all part of that white tapestry that holds the things that we hold dear together. We lose those, we're in trouble. Maybe not today, maybe not tomorrow, but we're in trouble.
This episode was reported by science journalist Veronika Meduna. She spoke to Sadie Mills and Dr. Rachel Pert of NIWA, and Kerry Walton, Andrew Stewart and Dr. Thom Lindley of Te Papa. This episode was produced by Veronika, with a little bit of help from me, Claire Kincannon, and Ellen Rikers. Sound engineering is by William Saunders, and Liz Garton is executive producer of Our Changing World. Our webpage is at rnz.co.nz/ourchangingworld, where you can sign up to our monthly newsletter. And if you've got feedback for us, you can email ourchangingworld@rnz.co.nz. Tēnā koe I whakarongo mai. Thanks as ever for listening to the show. Ko Claire Kincannon ahau. Have a great week. Kia pai, te wiki.